Okay. So we have moved into the functional territory, and I'm just curious how many folks are using resting state fMRI in their daily, day-to-day life. Okay, so yeah, a lot of people. So we don't really have to get into what it is and whether we believe it's real and all those things. So those are questions for 10 years ago. So basically, am I going the right way? Oh, okay. We'll have 1 minute as we get adjusted. I'll do some jumping jacks. Okay, all right. So we talked a little bit earlier about this idea of basically connectivity fingerprints, and this it comes from way back in our earlier sessions talking about anatomy and ways to define cortical regions. And I really like this Passingham paper. I think it's a 2002 paper where basically it talks about how the functions of a cortical area are determined both by their connectivity profiles, extrinsic connections, as well as intrinsic properties of that area, and that you can sort of define regions, in if you'd like to, by their connectional fingerprints, or how they're connected to other brain regions, and how that sets them apart from their neighbors. So, sort of continuing in this theme, we now have these new tools that resting state fMRI sort of gives us to characterize connectivity fingerprints or functional fingerprints in a very nice way. And if I could have, if you could just click play, perfect. So I took this lovely video from Daniel Margulies, our final speaker, because it just shows really nicely that you can get these spontaneous patterns of correlated activity across the entire brain, and they emerge, at a very slow time scale. But if you look at them over and over again, you start to see patterns. It's not just completely random, and it's not just completely spurious. Daniel, there he is. And so these, you can see things like language networks have their own sort of correlated patterns, and dorsal and ventral attention systems, and memory systems, and default mode networks. And basically all the things that we think about as functional networks can be characterized using these spontaneous correlations that we see in resting state fMRI, which makes it this really nice tool for many purposes, one of which we'll talk about today, which is brain parcellation. And I'm actually sort of an end user here of parcellation. I don't sort of go about creating parcellation schemes myself. I wish I had, because one surefire way to get a lot of citations is to create a parcellation scheme. So you may have heard of Thomas Yeo or Evan Gordon. These are all parcellation creators, and the papers get cited over and over again. And so it's a nice way to build a career if you're at the beginning stages. I sort of missed the boat, but it's not too late for some of you. So the goal is, for what we're talking about, intrinsic functional connectivity-based parcellation, there's many goals, but some of you may be interested in defining nodes in the brain for the purposes of graph theoretical analysis or other types of network neuroscience things. At this point, you can do whatever you want once you've created parcels, and I'll get into some examples of what you might do with them, but we're talking here mainly about how to get them in the first place. So the approaches that we've sort of gone over in the previous talks have to do with clustering by two different sort of main types of approaches. One is by voxel-wise time series similarity. So if you had something like 200,000 voxels in the brain, each with its own time series, you may group them together by the similarity of the time series, and you might, as you might examine, things that are spatially near each other might have more similar time series and might end up coming together in a clustering algorithm. And we talked a lot more about the other, I think, more common approach, which is clustering by functional connectivity similarity. So if you take, whatever your voxels are in the brain and create a correlation matrix of sort of ROI to ROI connectivity, or sort of voxel to voxel connectivity, and you group them together by their similarity in the functional connectivity profiles, that would be the method that we've seen a lot of, and I'll go over some of the approaches today. So I think one of the earliest studies, and I want to just make some comments along the way that are my opinion. I don't endorse any particular parcellation scheme. I don't sort of have a commercial interest in any one of these. But way back in 2008 is the first paper that I can remember that tried to use resting state fMRI data to do a whole brain parcellation. And back then, if you recall, we were often doing funny things like taking the rest blocks out of a long task scan and sort of concatenating the rest blocks and calling that resting state. So it wasn't even, back in the day, sort of pure resting state analyses like you do now, where you have someone in the scanner for 6, 10, or longer minutes. But in any case, that passed for resting state functional connectivity back then, and this was one of the first studies that actually took those rest blocks and said, can we actually use that data to do some kind of meaningful brain parcellation. So the goal here, and this is actually held up in later studies that used sort of real resting data from MRI data. But the goal in this first study was to identify locations on the cortex where the patterns of the resting state functional connectivity changed rapidly, representing putative boundaries between functional areas. And I won't get too much into the methods and stuff here, but in this particular case, they used this measure of eta squared to compare images instead of correlation, which, like a pure Pearson correlation, because the goal here was to quantify the difference or similarity of two consecutive images and not necessarily the correlational relationship between them. So this is just from the paper. It's sort of the outline of their general approach. And this was actually a pretty influential, I think the next one is, this is actually a better figure from the paper. But for example, if you look at these two parietal areas in the red and the blue, what basically you're doing is going voxel by voxel and looking at the whole brain functional connectivity of every voxel sort of along a line that would connect these two areas. And there's a point at which the sort of connectivity profile of the voxel changes from like one voxel to its neighbor, essentially. And that's what's being represented here in this sort of blue and red lines. There's sort of a crossing point. And the idea was that when these sort of connectivity similarities start to vary in this way, then we can infer the existence of some boundary between those two regions. Here it's SMG and angular gyrus of the parietal cortex. But the idea is mainly like when the functional connectivity profile starts to vary along the surface of the cortex, that's the point where we can say, okay, here's a boundary here. And we'll hear more, in the next talk about whether or not we should think of these, and we have already heard about whether or not there are strict boundaries, whether there's gradients. I think that's part of the fun of this workshop is kind of exploring both of these possibilities. But I'm just the messenger at this point, so don't shoot the messenger, essentially. So there's a follow-up paper to this approach by Wig and colleagues that sort of, took it to the next level, greater number of subjects. I believe this was all surface-based as well. And I tried to sort of reproduce these gradients that were, or these boundaries that were first shown in the Cohen paper. So I think this approach has gained a lot of traction, and there's a lot of fans of it. And what I'm really doing today is giving you a survey of what I think are the most widely used influential parcellations that have come about from the resting data from our literature. And, in discussion, I think maybe we can get into why some of these approaches have been more influential than others, and why some have caught on where others people have sort of forgotten about and left on the wayside. But my job here is just to sort of familiarize you with the territory. So Thomas Yeo, a Singaporean, has basically put out, I think, one of the most cited parcellation papers, in the business. And this was a really nice paper because it did a lot of things. It had, 500 subjects in the sort of identifying parcels data set, and then 500 subjects in a replication data set. So it really did a lot of things to mitigate our concerns about heterogeneity and individual variability and things like that. Surface-based registration, and there was in the final paper there were basically two sort of a coarse and a more fine-grained parcellation, one with sort of 7 whole brain networks and one with 17 networks, and they used a stability to measure to decide on the number of clusters, and they basically are defining in this paper cortical networks as sets of regions with similar profiles of cortical functional connectivity. So this is, I believe it was in Journal of Neurophysiology. This paper, I should mention, is something like 50 pages long. And if you read the paper, it's something like 10 studies all in one paper. It's really a tour de force. And I'll just kind of, show you some figures from it that basically these are the cortical areas that were utilized in the parcellation. And this is sort of, again, the discovery sample of 500 subjects, the replication sample of 500. And this is on the left, the 7 network parcellation, and the 17 network on the right. This gets back to what we've been talking about all along about how many, parcels or clusters or what's the right solution for any of these problems. And they're really, the sort of sad truth is there is no way to answer this question that's going to satisfy everyone. So, I mean, you might as well ask the question, how many brain regions are there? Because that's what essentially parcellation is trying to get you to. So I'll talk a little bit about this, in one of the later slides, but the idea is, like, what are you really trying to do in your study, and is it going to be better for you to have a really fine-grained parcellation to answer your research question, or is it really going to hinder you computationally if you have, an excessive number of brain regions that you're using in your calculation? So a lot of these answers are not very satisfactory. They're really sort of individual investigator-driven. But in this case, this is a couple parcellations that have I think, proven to be very useful in terms of answering cognitive neuroscience questions, which is why I think this paper is so widely cited. In one of the 7 to 10 studies that are encompassed within this paper, what he actually does is show these sort of connectivity profiles of individual brain regions within his parcellated networks. So as you could see in these polar plots, like the different labeled regions show functional connectivity with brain regions that sort of distinguish them from each other, which makes them sort of suitably considered individual clusters, in a way that is sort of nicely demonstrated by one of like 20 figures in this paper. But it's a really good one to take a look at. So in subsequent years, really what people did was say, okay, how can I come up with a better clustering algorithm or do something slightly different in order to define my clusters based on resting state fMRI data. And so a paper by Cameron Craddock came out in 2012, and this particular measure was based on the similarity of functional connectivity between voxels in the brain. And this particular study used a spatially constrained spectral clustering approach. And if you really want to get into it, we'll have to ask Cameron Craddock. But basically, it used normalized cut, which is a measure that simultaneously optimizes within cluster similarity and between cluster dissimilarity. So it's not just like, are the voxels in one cluster similar to each other, but are they sufficiently different from the neighboring clusters? And in this paper, it was very nice because he did a comparison with the AAL atlas, the Zillow-Eickhoff atlas, the Talairach, and the Harvard-Oxford atlases to say, compared to all these other parcellation schemes, how does the Craddock atlas, sort of measure up on these sort of similarity metrics? And, he's got, again, different levels of 50, a 200, and 1,000 ROI parcellation. And he actually also did a completely random parcellation. And I believe one of the surprising findings from the study was that the random parcellation was just as good as his part, if I remember correctly. That was, like I said, I'm just reporting the results here. But in any case, I mean, in some ways, I'll get to this later too as well, but in some ways you can do a completely random parcellation depending on what the goals of your study are, and you may not be any worse off than if you used any individual parcellation scheme. And why that may be is a question for discussion. So the other sort of one that comes up when you talk about resting state fMRI whole brain parcellation is a study by Shen and colleagues, came out in 2013. This was 79 subjects. I'm giving you a little bit of the details of, like, very vague details on the methods and the subjects that were used, because as you can see, it varies widely from study to study. And, like everything in the field of fMRI, the early studies were a handful of subjects, and the later ones, try to get into larger sample sizes to, address questions of heterogeneity. This one used did I get that right? K-way spectral clustering? That's what it says in the methods. And so this was a group-wise clustering that jointly optimizes the group and the individual parcellation. So we talked a little bit about this in the previous sessions about, whether or not things at the individual level sort of hold up at the group level. And I think this one was one of the first studies that really tried to optimize both. So, and they did not have a spatial constraint on this algorithm. And they also tested it on synthetic data, so it wasn't just empirical resting state fMRI data collected from humans. And this is basically one of the figures where they're showing, when you have synthetic data, the beauty is you know what the ground truth is. How many clusters there are is determined by you, and you can see how well the algorithm does in uncovering those. So they were just showing that their particular clustering approach was very good at uncovering the number of clusters in their synthetic data, as well as their empirical data. And again, here are the sort of whole brain clusters that emerge from this particular approach. And there's not necessarily a ton of overlap between any of the approaches and any of the results from the studies I've showed you so far. So, this is just another demonstration of the fact that when you ask the question of how many brain regions are there, you can ask it many different ways, and you may get many different answers depending on what you're trying to optimize and what your end goals are. This is one that is, I think, used a lot these days, the Gordon parcellation, which was worked out in 2 different data sets, pretty large data sets, using this new format, SIFTY. But basically, this one also, I think a lot of people now, when they're doing these parcellation studies, say, well, how well does my algorithm, explain the data compared to all the previous parcellations that have come out there? So you're very lucky if you started this in 2008, where there was sort of no one else to benchmark yourself against. But now, every paper you publish, you'll have to say, how does this compare to Gordon? How does this compare to Yo? How does this compare to all the others that came before it? So this was actually another extension of the Cohen and the Wig, sort of the boundary detection algorithm. And I believe there were some other criteria that they were using to assess the validity of the clusters here. But if you look again, so of course he did a nice comparison with all the clustering that came before him. So looking at, these are the power region of interest, parcellation, the Craddock, the Shen, Broadman, Yo, AAL, and basically, they tested all of these against the null model parcellation, and I think if you look sort of closely, you can see, where is his on the, well, I guess it's not, I don't actually see his, this thing is too small for me to actually read it from here. But if you look basically at sort of these metrics, he was trying to demonstrate how the particular approach that he was using was sort of, in some ways, more optimal than some of the other approaches that have come before. And so this is where, it gets a little bit tricky, because what really we're trying to get at the application on the user's end. So which one of these approaches are you going to use and why? And I think that, I can give sort of opinions at this point. Some considerations in real-world applications have to do with the size of the ROI itself and how homogeneous you want them to be. So smaller ROIs are themselves going to be more anatomically homogeneous because they won't take up as much brain space, and so you can assume that the voxels that contribute to a smaller ROI are, delineating one brain region, hopefully. ROI number, so the more you have, the more computationally intensive whatever you're going to do is going to be. So these are obviously trade-offs that one has to consider in whatever application that you have. So for me, as a person who does cognitive neuroscience, I kind of want to give you some examples of like where you would choose which scheme and why. And of course, again, I'm not saying that I'm right, I'm probably wrong, but these are just things that have been done and are continuing to be done. So I'm very interested, for example, in the frontoinsular cortex, and for those of you who follow this literature, you'll see that it's one of those regions that's activated in everything, from emotion, sort of the affective pain domain, to subjective awareness, things like interoception, bodily states, somatosensory processing, and of course high-level cognitive things like conflict detection, switching attention, and inhibition. So it's one of those brain regions that really begs for a parcellation, because maybe the problem is we haven't subdivided up these areas within the insula appropriately, and that's why we have a hard time characterizing the functional profile of the area. So if you look at some of the resting state fMRI parcellations that have been conducted of this this lovely area, you actually find some convergence across a couple studies. So there was a paper by Dean and colleagues in 2010 and another one by Luke Chang and colleagues in 2012 that suggested that if you look at the functional connectivity profile of every voxel in the insula and you use k-means clustering to say, group the voxels by which cluster they, or which connectivity profile they exhibit, you do tend to see this dorsal anterior insula subdivision, a ventral anterior insula subdivision, and a posterior insula subdivision. There's other insula subdivision papers using resting state fMRI that have shown different solutions, but this is one that I saw in 2 papers, so I thought, well, at least 2 papers have converged on this. The thing is, you can always keep subdividing things. Like, I think of this example because I see my parents do this all the time. So if we're using our fingers to count, we have 5 fingers, right? 1, 2, 3, 4, 5. You use those to count. But if you're from Bangladesh, you actually count the knuckles. So you go 1, 2, 3, 4, 5, 6, 7, 8, 9. And you have a lot more things to count. You just have a lot more, units to use, and I see my parents do this, and I don't know what they're doing. But the idea is that they think of the hand as not just 5 fingers. They actually, use each piece of the finger as a number. And so you can keep doing that in any kind of parcellation problem, right? You can keep subdividing an area further and further. The question becomes, is it meaningful to you to do so? So I know that the recent Glasser Nature paper, which we said we wouldn't talk about, but I have to say that I think there's 12 or 16 insula subdivisions from that approach, and whether or not they bias something in terms of understanding the functional profile of the insula is sort of yet to be determined. But you can always subdivide regions further. It's just a fact of math. So in this particular study, Chang, and then we later kind of follow this up with meta-analytic connectivity mapping. The idea is that you see these sort of reproducible sort of profiles where, like, the ventral anterior insula tends to be associated with emotional and affective processes, if you look across brain map databases, for example. And those regions of the ventral anterior insula tend to co-activate with limbic regions across a variety of tasks. If you look at the dorsal anterior insula, it tends to be more involved in those inhibition, sort of higher level switching, cognitive processes, and tends to co-activate with dorsolateral prefrontal cortex and parietal cortex. And if you look at what happens in the posterior subdivision, you tend to get the studies of pain, somatosensation, and things like that, and you tend to see co-activation in somatosensory cortices. So there's some kind of meaningful thing going on here when you describe the insula in 3 subdivisions. There may be more, like I said, but this is a place to start. Okay, so transitioning to a completely other, like I said, if you look at all these parcellations I've described so far, there's different numbers and different approaches, and you may say, well, I just want to do something data-driven. I really don't want to pick a side and pick a parcellation. I just want to take my data and ask it to spit out how many things there are in it. It's never that easy, but you do know that when you do independent component analysis, you can specify the numbers of components you want. And so many people do, something like 20 components because they want to see the default mode network or they want to see a particular network. But if you do a pretty high model order, like 70 or 100, you just get more and more subdivisions of those networks, and they fall apart into little pieces. But those little pieces, guess what? Those are now parcels, and you can use independent component analysis in that way just to parcellate the brain. So you can easily tell your GIFT toolbox or melodic how many components you want out of your data set, and it will give you 70, it'll give you 100, it'll give you 150. And those things that it gives you are now these spatio-temporally independent units that, if you like, you can use as your ROIs in subsequent analyses. You can use to create a connectivity matrix, a ROI to ROI. So it kind of takes a little of the guesswork out if you really don't want to pick a particular scheme to work with and you just want to get the parcels from the data that you have. This is sort of one relatively agnostic approach. Why might you do something like that? So we use this toolbox from Vince Calhoun to do things like dynamic functional connectivity, where once you get these ROIs out and you get a correlation matrix of the entire time series, you can, again, subdivide up the time series into little segments as long as you want. Well, not really, but there's some constraints there. And then again, you can have k-means clusterings to tell you like how many states there are and how long an individual or a group spends in each state and things like that. So you can compute frequency of occurrence of certain states or dwell time of particular states. So, this is all to say that, once you've done the parcellation, you're probably going to do many, many other things with the data that has now been parcellated. Sometimes you may choose something like independent component analysis simply because it's an integrated sort of toolbox put out by the developers. So like once you use ICA, it's easy to use this toolbox to continue to do the dynamic analyses I just described. You shouldn't do something just because it's easy, but I'm saying that if you wanted to you're starting out for the first time and you're interested in dynamics and you wanted to use a toolbox, then you might choose ICA as your parcellation approach just for that reason, just so you know. State transitions. So this is like, for example, something we did with a research scientist in my lab, Jason Nomi, where he used the Human Connectome Project data and, like I said, used independent component analysis to first parcellate the resting state fMRI data into lots of like tiny little brain regions. And then he was interested in relating like functional connectivity dynamics with individual differences across some different processes here. And basically he used k-means clustering, got 5 different states, and found that individuals that sort of had a greater frequency of occurrence of state 1 and less frequency of occurrence of state 5 actually showed the higher working memory scores as in the HCP data set. And something similar for a measure of cognitive flexibility, individuals who spent more time in state 2 and less time in state 3 relatively showed higher cognitive flexibility scores. So these are sort of just a little flavor of the types of cognitive neuroscience or clinical neuroscience applications that you may be interested in, and why you might want to do whole brain parcellation in the first place. So, the resting state functional connectivity-based parcellation that I've flown through here, has a lot of caveats. I think I've sort of already brought them up as we've gone along. We're always asking this sort of very ill-posed question of how many brain areas or clusters are there. That's always the question. And it's a hard one, as we've seen throughout. You could, define them in different ways. Sometimes you find convergence between a diffusion-based parcellation and a resting parcellation and a MCAM parcellation, but more often than not, you don't see convergence across modalities. So that's a surprising, maybe worrisome feature, but it's something to think about. There's considerations of whether you really want to use a data-driven versus a hypothesis-driven parcellation approach. And at the end of the day, all of them are sort of a little bit of a combination of both, because oftentimes you as a user have to put a constraint on the limit of, on the number of clusters and things like that. So there's really no completely data-driven way that I know of to do parcellation. Like you're always, I think one of the previous speakers mentioned, you're always going to have to think about what you want at the end of the day. There's no way out of it. You have to think about how many clusters you want or what solution is more optimal for the type of analysis that you're going to run. Usually you're not just clustering for the sake of clustering. You're trying to do something then with the parcels that you've derived, so that what you're going to do after the parcellation is over is often really what drives your choice. And there's, you may be more concerned with some of these factors than others, like validation of the clusters, stability of the clusters, reliability of the clusters. It's funny, in my own sort of foray into parcellating, we did some studies where we compared like insula parcellation in autism versus typical development, and we used stability of the clusters to determine whether or not they were real and things like that. And I found some differences, but it's really hard to quantify. Like if you have two groups and they've clustered some brain areas, and now you want to compare those two groups on how different are the clusters in the two groups. There's no sort of real metrics. You can look at the cluster extent or the cluster stability or things like that. But long story short, we never published the paper because we couldn't figure out a good way to compare the two parcellations in the two groups. So it's one of those ongoing questions that the answers are not forthcoming. At least they're not straightforward. So I'm happy to leave time after giving this vague and ambiguous set of recommendations for any questions.